Ever wondered why molecule clustering plays such a pivotal role in drug discovery? It's all about understanding the chemical diversity of compounds and identifying potential drug candidates. And one of the most powerful tools used in this process is called self-organizing maps, or SOMs. First off, what's a molecule in the context of chemoinformatics? It's typically represented as a set of atoms and bonds. These representations are then transformed into numerical vectors that encode the structural and chemical properties of the molecules. These vectors serve as the input data for the SOM. Now, picture a SOM as a grid of artificial neurons organized in a two-dimensional layout. Each neuron has a position in the grid and is associated with a weight vector. During the training phase, the SOM learns to map these high-dimensional molecular representations onto this two-dimensional grid, all the while preserving the structural and chemical relationships between molecules. The training process uses a method called competitive learning. Each molecule is associated with the neuron, or a group of neighboring neurons that most closely matches its structural features. As the SOM is trained, molecules with similar structural and chemical properties tend to be mapped to nearby neurons on the grid. This results in clusters of molecules where compounds within the same cluster share similar characteristics. And just like that, we've got a visual representation of these molecular clusters. This visualization helps chemoinformatics researchers and drug discovery scientists to understand the relationships between compounds much more easily. So, how does this apply to drug discovery? Well, by clustering molecules based on their structural properties, researchers can identify clusters of compounds with similar structures. This can lead to the identification of lead compounds that have the potential for further development as drug candidates. SOMs also assist in analyzing the chemical diversity of a compound library. This ensures that the library covers a broad range of structural diversity, which is crucial for effective drug discovery. What's more, SOMs can be used to identify relationships between molecular structures and biological activity. By analyzing the clusters, researchers can gain insights into the structural features that influence a molecule's bioactivity. The clustering results can also guide virtual screening, where computational methods are used to assess the potential of compounds in a library to interact with specific biological targets. Finally, in the process of designing new drugs, chemists can explore the clusters to identify molecules with desired structural features and optimize their design based on the clustering results. In a nutshell, self-organizing maps are a valuable asset in chemoinformatics and drug discovery for clustering and visualizing molecules. They help researchers identify potential drug candidates, understand the diversity of chemical structures, and design new drugs more effectively.